a good slime mold, right? So this is the concept behind why we need to study transcriptional regulation. Slime molds are studied by geneticists, developmental biologists, and others. By the way, those are the red objects, the slime molds, not the moss that's behind it. Slime molds do this really crazy thing. They're initially single-celled organisms. They are individual single-cellular organisms. They're like amoebas. Right? Each cell is its own organism. They can move around. They can take up nutrients. They reproduce and so forth. But they reproduce asexually. They divide and produce daughter cells and so forth. But if they're in a situation where there's a lot of crowding, a lot of amoebas, single-cellular organisms around them, or if food starts to run low, then all of those amoebas start to stream together. They crawl together into a big mound. They become a multicellular organism, essentially. Some of those amoebas, those individual cells, basically give up the ability to have sex and reproduce, and they form a stalk, like a plant stalk, right? A tall structure that builds from the ground and lifts up what you see here, those red parts, those are the fruiting bodies. So in that population of single-celled organisms, when they come together, some of them decide, I'm going to become a stalk, and I'm not going to reproduce anymore. I'm just going to help my friends, the other amoebas. I'm going to lift them up, and those ones that are in the fruiting bodies then can sporulate. They can actually reproduce. And the idea, supposedly, behind that stalk, lifting the fruiting body up, is that a passing insect, mammal, whatever, might rub against it and transfer some spores somewhere else where there might be more food or nutrients where those colonies can start growing again. So this is studied because it's an excellent transition in developmental biology, because this is a single-celled organism that can also decide to become a multicellular organism, like one of these stalks with a fruiting body on top. And these cells, the point for genetics is somehow these cells decide to adopt one fate or the other. When it's time for these fruiting bodies and the stalks to form, how do the cells know which ones are going to be stalks and which ones are going to be fruiting body cells? And most importantly, all of these cells have the same genomes, because it's all the same species. They all used to be amoebas, but they've changed. That's transcriptional and translational control. Because all of these individual cells have the same genes, it's whether or not you turn on the genes that make you stalk and turn off the genes that would make you a fruiting body that makes you a stalk cell. And if you turn off the genes that would make you a stalk cell and turn on the genes that make you a sexually reproducing cell that's in the fruiting body, then you've got a different type of fate. And that's what's called genomic equivalency. That different cells in our bodies have the same genes, but by turning some on and some off, that's how we get to be so complex. One of the items to watch in the movie for today had to do with consensus sequences and understanding where proteins bind in a DNA sequence. And that was illustrated here in this video, where we have some yellow nucleotides. These are a bunch of different promoter sequences for different genes. Each line represents the promoter, the sequence of DNA upstream of where transcription starts, for a bunch of different genes from E. coli. And so, as I discussed in the movie, you line them up like this and you look for common patterns. Those are the highlighted nucleotides. And that's how scientists decide, on average, that's about the proteins, or the DNA sequence that a protein will bind to.
Are you recording?
Yes.
Okay.
Thanks for checking, though. I appreciate it. So that's how scientists came up with these consensus sequences. It's an average. Now it's your turn to do this. So take a couple minutes. These are totally fake. I just made this up. But there's a consensus sequence buried in here somewhere. So you do the same thing. I've lined up all of these promoters, supposedly, from different species, promoters of the same gene, say. So see if you can find if there's an average DNA sequence in here somewhere. Make the consensus. Take about 1 minute to do this. If you end early, make sure you've completed the Socrative quiz. Raise your hand if you have any questions. It's awfully quiet. What about the first column? Is there a consensus for the first column of nucleotides?
In A.
I'd say no. You're looking for, in every column, the most frequent nucleotide, but especially if it's mostly one nucleotide. So in column A, I wouldn't conclude that there's any particular agreement or consensus.
Well, there's more A's than others.
Yeah, but there are not that many A's.
So,
3 out of 10? 3 out of 8? Not enough.
okay, so not every column's gonna have.
Yeah, not every column is going to necessarily have a consensus nucleotide. Somebody found one yet?
No, it's a question.
Yeah, please.
I realized that in the video, what you put the most was T and A's on the 35 and the 10 binding.
Yeah.
And well, that also counts as a consensus sequence here. Is there like reason we learned before that normally the place where the RNA polymerase binds is full of G's and C's, and is it just a coincidence that there were a lot of T's and C's?
Basically, yeah. It doesn't, yeah, the reason
So it could be like G's and C's.
that there are more A's and T's in the minus 35 and minus 10 sequences seems to be a coincidence. Yeah.
In column 7, I think it's 7.
Let's see. 1, 2, 3, 4, 5, 6, 7, 9, 11, 13, 15, 17. Okay, so column 7.
Yeah.
8 and 9.
So in column 7, we've got 1, 2, 3, 4, 5, 6 out of 8 A's. And then there's a bunch of C's in the column next to it. What next?
Bunch of A's in the column next to that.
A's,
T.
6 T's in that column, and then we go back to, there's 3 G's. That's not, we're looking, say, for more than 50%. That's not a true genetic threshold or cutoff, but for this purpose, let's say 50% or more. So it looks like if there's a consensus sequence in these promoters, that would be it. And that's how biologists would say, in general, proteins that bind this promoter sequence are going to recognize the sequence a cat. One of the longest sentences you can create using the 4 letters in the nucleotide code. Okay. So that's the concept of a consensus sequence and how scientists develop them. It's a figment of our imagination. They're from real data, but the concept of the consensus sequence is it's an average. Yeah.
So what you did was you took the most occurring one.
Correct. So in this column of nucleotides, there are more A's than anything else, and at least 50% of them are A's. So it seems like on average, when a protein binds this promoter sequence, it's recognizing something specific about each of these sequences. Did any of those sequences actually have the consensus?
You mean all of them had the same
Oh,
one?
yeah.
Yes, exactly. Was the consensus sequence actually a real sequence that existed in one of these species? Just one. I totally didn't do this on purpose, but honestly. But it turned out that the cat was the one where a cat was part of the DNA sequence. So again, the point is that proteins have some give to them. Just because a protein is specific to binding a particular gene's promoter doesn't mean it has to bind to the consensus sequence. It'll bind something that's sort of like the consensus sequence because the consensus sequence is an average. It's just a useful way for geneticists to say, in general, proteins that regulate this gene will recognize a sequence that looks something like this, ACAT. Even, yeah.
Is there a guideline for the number of repeats that would make it, say, more recognizable by standard? Like they want a certain number of repeats? Because I mean that's a relatively short sequence that it could be construct to anything.
So a couple of points. Repeat sequence not necessary for binding of transcription factors. So transcription factors don't necessarily bind the same sequence over and over again. But it's an interesting point that this is only a 4 nucleotide sequence. And we've looked, I think we've done the math before, how frequently 4 nucleotides in a row will show up in the genome. So how frequently do you expect to find a 4 nucleotide sequence? Probability of getting an A at the first position is 1 in 4. So the probability of getting those 4 specific nucleotides in a row is 1/4 to the 4th power. Oops. I don't know what that 6 is doing there. Forget about that. Yeah, so 1 in 256 nucleotides. Which must raise a question in your heads, I hope. Wait a second. If things like TATA binding protein, eukaryotes now, that recognize the sequence TA, that's going to be every 256 base pairs in our billions of base pairs in our genomes. So TBP then, these transcription factors or sigma factors in prokaryotes, they're not the only thing that controls transcription. Because if they were, if all you needed to know was 4 bases in a row, like a cat, to know that's where a protein is going to bind to initiate transcription, then everything in the genome would be transcribed, almost. Genes would be everywhere, which isn't true. So there are other proteins that are involved as well. Thank you. All right, let's check out Socrative. One gene encodes 1 protein. That's a negative. Not every, it's true for some genes, but not for all genes. In some cases, 1 gene will encode multiple different proteins. Ooh, I heard some people, I'm glad that you discuss these things when you're waiting before class. I'm glad to hear those conversations going on. Ah, fantastic. You weren't crazy. There are 20 naturally occurring nucleic or amino acids. So 20 is the correct answer for that. Good. That means we have a lot of stuff we don't have to talk about in class. Ooh. Similarities between nucleic acids and proteins. Yeah, they're all part of the central dogma. DNA makes RNA makes protein. Are all of these? I think all of these are true. Wait, there's one that's not true.
Is it D?
There's one of these that's not true.
Is it D?
It's B, as in go Bulldogs. Both. So which one? What molecule is not read by an enzyme to produce yet another type of molecule? So DNA, what central dogma? What reads DNA? What enzyme? What's the molecule that's produced after an enzyme reads DNA? DNA makes RNA. That's central dogma. DNA makes RNA makes protein. So DNA is read by RNA polymerase. RNA polymerase produces RNA. Then what reads the RNA to produce the protein? Ribosomes. And then what reads the protein? Nothing. Because that's the end of the central dogma. DNA makes RNA makes protein. And that's why B is wrong, because proteins are not read by some other enzyme to produce some other molecule. Protein is the end of the central dogma. So anybody have any questions about D? D is true. Both DNA, RNA, and proteins are all made of strings of subunits. Okay, so what do I mean by linear strings? Well, how do you write out the sequence of a DNA molecule? Somebody tell me the sequence of a DNA molecule. Make it up. ATGCT, I don't know, CA. It's on the video or the audio recording. You can go back and correct me if I'm wrong. Okay, so that's a linear string of subunits, each subunit being a nucleotide. Same for RNA. What about a protein sequence? Right, have a nice day. They're linear because they don't branch. Yeah.
You cannot see the screen.
Uh-oh.
Sorry.
Don't be sorry. I'm sorry. Yep. So they're linear strings because they don't branch. So you don't have any DNA molecules where there's a spot at which that T is also linked to a G, A, C, T, and proteins don't do the same thing either. You don't get a spot where that happens. There's no branching in nucleotide sequences or in amino acid sequences. Other questions?
Well, since we're talking about amino acids,
Sure.
20 are essential, but there's 23 that you're supposed to use.
Oh, there are a bunch of weird amino acids. Yeah. There are 20 essential amino acids. Yes, there are. So maybe I was wrong. I will take that challenge. Are there 20 naturally occurring amino acids or 20 essential amino acids? I was probably wrong to say 20 naturally occurring.
Like 500.
Well, in my hometown in Oregon, there used to be, I think there might actually still be a company that synthesized amino acids for biotech companies. And they made all sorts of crazy synthetic amino acids, but they were still amino acids. So, not essential amino acids, but there are more than 20 amino acids.
So,
The essential ones, there are 20. We have to have 20. 20 make up the proteins in our bodies. And then if you want to get into advanced genetics and more biochemistry, you can talk about all the other amino acids and their craziness. But not here, not here. Let me grab this photo really quickly so I can edit it. How did you figure out the number of amino acids? How do you know how many amino acids are in here?
well, you can look at the carboxylic groups and the double bonded oxygens.
Okay, so what I'm hearing is, what's the bond that bonds amino acids together?
C to N.
It's called a peptide bond. That's this thing. So the real question is, how many of those are there in this molecule? Looks like 3. And those are between amino acids, so how many amino acids are there? 4. So I hope you didn't pay attention to the coloring, because I didn't do that on purpose. I just grabbed this image off of Google Images, but I thought, oh, that's kind of nice. Maybe it'll throw some people off. So the important thing to recognize there is that peptide bond, the things that circled. Okay, that separates the amino acids. Does anybody know which amino acids these are?
These two do.
Whoa. All right, what's number 2? Glycine. Number 3? Uh-oh. Well, maybe let's flip ahead. Everybody look on the next slide, or whichever slide I put the amino acid table on, for number 3. Okay, fine, it's serine. Okay, everybody look for number 4. What's the chain that differs between all these? What's it called? Side chain, also called R group. Okay, so here are the R groups. Glycine is just a hydrogen. Number 3 we said was serine.
4 is alanine.
Yep, 4 is alanine. Its side chain is a 1-methyl group.
And that one, is that what's called? Is it valine?
So that one's got,
Is that valine?
I think so,
Okay.
valine. So 4 amino acids. So we're going to get into this process later of reading messenger RNA and producing proteins. That's translation. That's a class that's coming up. We're almost there. Let's see. So, writing protein sequences. Oh, it's not 5' to 3'. That's notation that's just for DNA and RNA. So we do N-terminus to C-terminus, which is the same thing as amino to carboxy. So both C and E are correct here. They mean the same thing. The N-terminus is the amino terminus because an amino group has what atom? Nitrogen. And the same is true for the element next to it, C, carbon, carboxy terminus. So C terminus, that's equivalent to the 3' end of a DNA molecule. So that's the important concept here, is that DNA is written 5' to 3'. RNA we read from 5' to 3', and the protein that's produced from that RNA molecule is produced N-terminus to C-terminus. So 5' in nucleic acid and N-terminus in protein language are equivalent, but you can't say the 5' end of a protein. And you can't say the N-terminal end of an amino acid, or DNA, RNA. That doesn't make sense. Any questions now about any of these questions? How, are you feeling confident about this one? Ooh, that's close. So you're saying it's either 3 or 4 amino acids. We have to get through transcription. So I'm going to leave you to think about the answers to these questions here. I'll show you the answers to this question, just so you've got them in the recording. So there you go. The names of the amino acids, their single letter codes, their 3-letter codes, their full names, and then the answers to those questions. So you should become familiar with how to read amino acids in the single letter code, the 3-letter code, and know their full names. Because if you haven't done this by now, you'll encounter this in maybe organic chemistry, definitely biochemistry. You'll use it again in cell biology, if you're a biology major. So it's valuable to start taking the time to get to know the amino acids. That's how you spell "have a nice day" in amino acids. Okay. I'd like to tell you briefly about my favorite protein, tau-binding protein. The only reason I really like this protein is because I worked on it when I was in your space as an undergraduate when I was a student taking genetics and biochemistry. My first research experience was working with this protein, which, as you've seen, binds to a DNA sequence, TA, and this is a picture of TBP bound to DNA, well, drawing. So here's the double helix, and then the ribbons and the alpha helices here, and those beta sheets, the long strips, are the structure of TBP. So TBP, like most, this is the critical point here to remember. Proteins usually have two different domains. They'll have a protein-protein interaction domain, and they'll have a DNA binding domain. So if it's a transcription factor or any sort of protein that binds to DNA, binding to DNA itself usually isn't enough for the protein to actually regulate genes being turned on or turned off, transcribed or not. They usually work by A, yes, they bind the DNA, and then B, they have to interact physically with some other proteins. Remember slime molds. They've got tons of genes in them. Those genes need to be turned on or turned off, but it isn't enough for TBP, taught binding protein, to bind a 4-nucleotide sequence, because that's going to happen 1 every 256 base pairs. So it has to interact with other proteins that help regulate transcription. So that's that. And this is just a more complicated example than what I present in the videos for this semester. It isn't really truly the case that TBP, which is shown here, directly binds to RNA polymerase and brings it to transcribe the gene. It actually works through a series of other proteins that connect it to RNA polymerase. So I'm just trying not to burden you with too many of the details because there's a ton of proteins, as you can see, that are involved in actually initiating transcription. Right? The basic point is transcription factors bind the DNA first in a sequence-specific manner. Those transcription factors look for a specific DNA sequence, the consensus sequence. Then they connect with other proteins that help actually start the process of transcription. And this might go over well at Fresno State, I'm not sure. The structure of TBP reminded people of a saddle, except I can't spell stirrups, but this structure of the protein reminded the crystallographers of a saddle with stirrups, 2 loops of the protein sequence hanging down on either side. And what this space-filling model represents is, most of this is TBP, but here, these sticks that I'm circling in blue are DNA. So most of what you're seeing there is the structure of the protein. But this is a crystal structure where scientists actually look at where the atoms in the TBP molecule are located relative to DNA molecules. So this is TBP bound to DNA, the sequence that it likes to bind to, TA. And you can see, hopefully, maybe zoom in on your tablets if you can't see up here, that the individual atoms in that DNA sequence fit really nicely and pretty snugly around the molecules and the atoms that are in TBP, right? This surface where the protein is actually contacting the DNA molecule. It's like Legos. They fit really tightly together when that protein is bound to the DNA sequence. And those 2 amino acids that are highlighted there, it's hard to read. That's Lu205, which is also abbreviated L205, and isoleucine 194, which is also in single letter code I194. So those are 2 specific amino acids in TBP. What do the numbers mean, do you think? 194 and 205. Number of amino acids. There are 194 isoleucines. It's location. Starting from where? The N-terminus. So if you write out the full sequence from N-terminus to C-terminus of TBP, when you get to the 194th amino acid, it's normally isoleucine. And when you get to the 205th amino acid from the N-terminus, that would be amino acid number 1. Then the 205th is leucine. So that's all that nomenclature means, but it's important to know this nomenclature. When you see a single letter followed by a number, you're talking about a protein that's positioned in the amino acid sequence of that amino acid. And those two amino acids are critical for telling TBP or showing TBP what DNA sequence to bind. Because those are the two amino acids that make those really tight interactions with the DNA molecule. Half a minute, 45 seconds. Talk to your neighbor about this question. If you were going to have some sort of a color code that represents the nucleotide-nucleotide bond, so A to T has that particular sequence of 4 colors, and T to A in a different sequence, which of these codes, the panel on the left or the panel on the right, would you rather have if you were a protein, you were trying to figure out what DNA sequence you're binding? What do you think? So it sounds like there's some discussion. Some people have already reached consensus. So what's the debate? Okay, who thinks left side?
Oh, Riley's left side.
Like 3 or 4 people. Who says right side? About the same number. So argue it out. Left side has more information? Just because it's got more colors?
It's because the left side has a lot more rare, a lot more different differences in it, so it has to be very specific for us to find it. So it's less likely that we will make a mistake in actually locating that particular sequence.
Right. So the problem with the right side, it's the left side wins. The problem with the right side is if you were a protein and you were looking at this sequence through this color code, which is not, of course, what happens. Proteins are colorblind. You wouldn't be able to distinguish as a protein an AT base pair from a TA base pair because the colors are the same, the pattern's the same. And the same problem you have for GC versus CG. So if you were a protein and you were reading the 5' to 3' direction and you saw red, green, red, that is green in the middle, right?
Gray.
Gray?
Gray.
Crap. Is it red?
Yes.
Okay, good. So if you saw red, gray, red, you wouldn't know if you were reading an AT base pair or a TA base pair. And the same with GC. You wouldn't know if you were looking at GC or a CG. So if you're a protein and you're trying to read the sequence of a DNA molecule from 5' to 3', A and T look the same to you using this code. That code, of course, is an abstraction, but it tells us exactly about what the proteins do when they're sensing, when they touch the DNA and they're sensing what the sequence is that they're looking at or they're interacting with. So what is the information, not color? What does color represent? What is the information that proteins can use to feel what sequence they're interacting with? It's the molecule of DNA. So what about the molecule of DNA?
Like the slight differences between adenine and thymine and stuff like that.
So it's subtle chemical differences between the 4 nucleotides. So those colors represent different bonds that can be made between the protein, electrical interactions, the way proteins feel, between the protein and the nucleotides. So if you look in the major groove, for example, of an AT base pair, then those colors represent different bonds that can be made hydrogen bonds, electronegative, electropositive interactions between proteins and the edges of those base pairs. And each AT and GC differs in the order of, what's red representing? Right, so red, there's a pair of electrons there on nitrogen. This is why you have to take organic chemistry for genetics. There's a pair of electrons there, so that's an electron donor. Blue, there's a hydrogen that can form a hydrogen bond with a protein that's touching that piece of DNA. What about oxygen? It's an electron donor as well. Right. So there are positive and negative charges along the edges of the nucleotides, and that's what proteins feel or interact with that makes proteins stick more to one DNA sequence than another.
Do we not have that image?
You do not have that image. You do now because it's in the recording. And likewise, now you have, oh dear. Let me try that again. That was ugly. I want to give you this image too. Because maybe it gives, I don't know, hopefully it's helpful to you. It gives a better sense of the differences between the edges of the nucleotides. So when you look at a DNA double helix straight on, you see the base pairs as flat objects. But if you pull one of those out and you flip it so you're looking at it from the top down, like this, then you can see that there's a difference in AT bases than GC bases on the edge. And that's the edge that a protein can interact with, because a protein's coming at the screen like this. It's going to bind to the DNA molecule. It doesn't feel the insides of the base pairs. It doesn't feel where the base pairs are hydrogen bonding with each other. That's in the middle of that double helix. The only thing that proteins can touch or sense is the edge of the double helix. So that's the information along the edges that proteins can use to sense what is the sequence of DNA that I'm actually physically touching. And it turns out, I didn't point this out on the slide, I should have, a couple slides back, the major groove, the point of that whole color coding thing, the major groove has more information about the individual identities of the nucleotides than the minor groove. So the minor groove, the panel that was on the right where the colors looked the same if it was AT or TA, that's why most proteins do not interact with the minor groove of DNA, because there's no information there that helps them figure out what DNA sequence they're interacting with. The major groove's where all the action's at. Get your major groove on. Okay. TBP, on the other hand, is kind of weird. TBP is a weird protein that interacts with the minor groove. It's really weird and really rare for proteins. It's a very small class of proteins that actually interacts with the minor groove. Maybe that's because it's looking just for a sequence that has just A's and T's. Right? We just saw that A's and T's in the minor groove, they look different than G's and C's. So maybe TBP doesn't really care if it's ATA-T or TATA it's binding to. It just looks in the minor groove for that specific set of signals from the nucleotides. Now, because I love TBP, I put a lot of information in here about TBP. Look at that. We just skipped right past it. That's fine. I didn't know I was going to go off on you about getting your groove on, but it happens, and that's okay. So don't worry about that material. I know it's in the lecture slides. It's okay. So yeah, we'll talk about reporter genes later. So here's an interesting final thought to keep you going over the weekend. Because you need genetics for the weekend, so don't fade out on me yet. We've got, I know it's time, but thanks for hanging with me for literally 1 minute. Here's us, humans. We have a huge genome size in terms of millions of base pairs compared to all of these that are across the bottom. Plants, worms, flies, etc. They have tiny numbers of tiny genomes. What makes us so much cooler than all of these single-celled organisms, plants, and other boring organisms? We're complex, but you don't have to be complex by having a bigger genome, right? Look at Arabidopsis. We've got a huge genome, but we make so many more proteins because of splicing. And that's what we're going to start talking about next class. We're going to learn about maybe whether or not splicing is regulating the different colors that you see in African cichlid fishes that live in Lake Malawi. So to prepare for next class, by the way, I'm going to send you a Google Classroom assignment right now. That's that exam 3 question. It's due Monday so that I can compile it and give it to you to study for the exam. 2 videos to watch, and 1 reading assignment that's already in the syllabus. For Monday, have fantastic weekends. Thanks for your attention. Come on up.